Dear audience, in this talk I will present to you our lab, our laboratory of immunology at Migal, led by Dr. Weinstein-Marom, a senior researcher in the lab, and by myself, Gidi Gross or Gidi Gross. I will describe to you our field of interest in the lab, and this is Immuno-gene Therapy. Immuno-gene Therapy means that we design genes, we create new genes with which we attempt to instruct immune cells to treat disease. And how do we do that? The two different types of cells we mainly work on are the killer T cell, a T cell in our body that is geared to kill infected cells with viruses or bacteria, or malignant cells, cancer cells. And the regulatory T cells, these are cells that are, their role is to protect our body from overreaction of the immune system, from autoimmune responses, autoimmune diseases, and inflammatory responses when they are, where they pose danger to the body. So, what do we try to do with these cells? So with a killer T cell, we try to identify new targets and new means to direct these T cells to attack cells that express these targets in a very specific manner. We create logic gates, which I'll describe to you a bit later on. We create genetic switches, namely we can control the time and manner in which the T cell would come to work. And we create new CARs, chimeric antigen receptors, that I will explain to you in a minute. We create genetic adjuvants, namely these are genes with which we help the T cells, either regulatory T cells or the killer T cells, to survive better, to survive for longer times in the body, and to work in an enhanced manner. And there are different parameters that these adjuvants are directed to work on. And in the Treg world, we attempt to create, by genes, using new genes, a new source for regulatory T cells. We can convert regular T cells, normal T cells, into regulatory T cells, and so that we can expand these cells to large amounts with high purity and stable functional properties. And the diseases we are attempting to treat with these regulatory T cells are inflammatory bowel diseases, namely Crohn's disease and ulcerative colitis, celiac disease, type 1 diabetes and other autoimmune diseases, and atherosclerosis. So more than 30 years ago, when I was a PhD student at the Weizmann Institute of Science in Rehovot in Israel, under the supervision of Professor Zelig Eschar, we created the first the prototype of the double-chain chimeric antigen receptors, or CARs, and the first prototype of the single-chain CAR, which is the basis for almost all CARs that are today in clinical trials, in clinical use, and in advanced preclinical studies. Today, 3 CAR T-cell therapies have been approved by the FDA to treat B-cell malignancies, leukemia, and lymphoma of B cells, and close to 670 CAR T-cell trials have been registered as of mid-2020 all over the world, trying to treat either hematological malignancies, namely leukemias, lymphomas, multiple myeloma, and many, many solid tumors. But success, especially for the solid tumors, has been rather limited. And one of the challenges is actually finding the right antigens, the right targets. So how can we tell a tumor cell from a healthy cell and target these tumor cells without affecting the normal cell by a CAR T cell? And so why is that a challenge? Because tumor cells do not normally express antigens that can be targeted by CARs, so that one CAR can be useful for many, many patients with perhaps different types of tumors, because those antigens are usually expressed by other cells in the body, many of which are essential to our health. So when the engineered T cell that expresses this CAR would target the tumor cell, it would also damage healthy tissue and, in fact, would cause a life-threatening risk as in doing so. So how can we prevent these CAR T cells to attack normal tissue but still attack cancer cells? So here we try, this is one of the major arms of our research today in the lab. We try to do this by implementing the principle of logic gates, which is taken from the electronical or computer world. And this is, a logic gate is an apparatus that can translate, according to the blueprint of instructions, different inputs, binary inputs, either absences or presences, into a binary output. And in the world of a T cell, of biology and a T cell especially, this would mean that a binary yes or no would be above or below a certain functional threshold. So we're talking about either presences or absences that can be incorporated into the form of inputs so that a certain type of receptor could translate these inputs into an instruction for the cell, kill or do not kill. So the simplest logic gate would actually be the one that functions in normal T cells or in CAR T cells. Namely, there is the antigen in the right amount, and the cells feel this presence of this antigen and kill the target cell that expresses this antigen. Now, if there is a situation in which a tumor cell or the tumor cells in a certain cancer patient are the only cells in the body that co-express antigens A and B. So other cells in the body can express either antigen A or antigen B or none of them, but no other cell in the body expresses A and B. If we can find such pairs, and we can then implement the logic AND gate that is sketched here to allow T cells to target only cells that co-express antigen A and B, as shown here in the sketch. So in the left-hand side, we see a T cell in the upper part that has two receptors, but only one of them is engaged because that cell expresses only antigen A. Only when the target cell expresses both antigen A and B by a certain cellular apparatus that is a product of a gene, would the killer cell kill the target cell that expresses these two antigens together. So this is one example of a logic gate. This is logic AND gate. Another example, not shown here, but we work along this line as well, is the logic AND NOT gate, which instructs T cells to work only when one antigen is present and another antigen is absent, which is a good way to protect healthy tissues that expresses the antigen that is not expressed by tumor cells. So, in fact, this world of logic gates poses two major challenges. One of them is identifying the right cues, the combination of cues that can serve as targets for genetically engineered T cells. And these cues should not necessarily be antigens or targets that are expressed on the tumor cell, but also elements that are present or absent from the environment of the tumor. So these are environmental cues. So which are the right cues? Which are the antigens and the cues that are present or absent from the tumor that actually form a special signature that is unique only to the tumor cells and not to any other cell or other tissue in the body? And the other challenge is, since now that we have these cues, how could we target them? How we can translate this knowledge into a means with which we can equip T cells to kill or avoid killing, depends on the context. So these two different challenges we are engaging now. We are creating in Migal, in our lab, new types of receptors with which we hope to achieve this ability to translate the signals of presence or absence into act or do not act. And in Impact Bio, which is a new company, not so new already, we're trying to identify, and we have actually identified a new landscape of such signatures, which we hope we could use in the future to target tumor cells specifically and spare and protect normal cells in the body. Now, in the Treg world, which is the other arm of our research, regulatory T cell or Treg, we are trying to achieve two different major goals. One is how to target regulatory cells cells that are supposed to suppress the immune system, to inhibit the response of the immune system, in those cases in which the immune system, by mistake or by another fault, actually creates a disease, an inflammatory disease, such inflammatory bowel diseases, celiac, all sorts of autoimmune diseases, atherosclerosis, allergies, etc., etc. And today we know that overreaction of the immune system is one of the major problems that leads to morbidity and sometimes to mortality in the coronavirus pandemic. So how do we specifically suppress an undesired immune response while leaving the rest of the immune system intact and fully functional? So we're trying to identify new targets specific to these diseases and others, and to design the right receptors to target these new entities, either on the cells or other structures that are specific to the diseased tissue in those diseases, inflammatory diseases. On the other hand, we are engineering normal T cells of a certain type to become regulatory cells and to remain as such for long periods, namely they would be stable, and we can expand them, and we can make sure that they are stable so they do not turn into effector T cells, but rather remain regulatory T cells that can suppress inflammation as desired. So this work on regulatory T cells, we are now joining efforts with a new company called GentiBio, and we have other partners from the US. We all attempt to achieve these goals, namely to engineer cells to become high quality and high amount of regulatory T cells, and to target these cells for treating all sorts of inflammatory diseases in a very specific manner. So here I summarize two different arms of our research in the lab, namely fighting cancer with killer T cells and fighting or treating inflammatory diseases with genetically engineered regulatory T cells. And this is our team in Migal. Thank you very much.